let me introduce first speaker for our session is Bryn Taylor. Dr. Bryn Taylor is scientist in Silico Discovery Group in Johnson & Johnson Innovative Medicine. She got her PhD in Biomedical Science in the University of California, San Diego in 2020. She works on early stages drug development for difficult targets in oncology. We focus on using computational tools to advance modalities such as target protein degradations. Taylor, the stage is yours.
Thanks, Alexei. Okay, I'm going to go ahead and share my screen. And slideshow. Can you see the correct screen?
Yes.
Great. Okay. Thanks very much for the introduction, and thanks to the organizers for having me here today. So as Alexei said, I work at Johnson & Johnson Innovative Medicine in the In Silico Discovery Group in La Jolla. And today I'm going to talk to you a bit about what we're doing to understand PROTAC permeability using molecular dynamics simulations, Markov state models, and deep learning. With that, we'll get started. So as a brief background, PROTACs, or proteolysis targeting chimeras, are heterobifunctional molecules that enable targeted protein degradation. So they are as I said, they have two warheads and a linker. So one warhead will bind the protein of interest, the POI there, and the other one binds an E3 ligase. So the E3 ligase will then tag the POI with ubiquitin, targeting the POI for degradation by the proteasome. So this is in contrast to the classic small molecule inhibitor approach for targeting a protein and simply removing its function. This will instead degrade the entire protein altogether. So as you might immediately guess, a very common issue with using PROTACs as drugs is simply that they're really large, complex molecules, which have low bioavailability, and that may in part be due to their low permeability. So what I'm showing here is from a great paper assessing the PROTAC degrader field in industry. So my former colleague, Lori Volak, surveyed scientists at several different pharmaceutical companies and asked them to report what were the most significant ADME or physiochemical property challenges facing development of orally bioavailable degraders. So the plot below shows that in blue, those bars are the primary challenge, and in red is the secondary challenge. So this was just all voting from people surveyed, but people pretty much agreed that it was permeability and solubility were primary challenges, along with molecular weight, which I think does contribute to permeability and solubility. And secondary challenges, these problems are very complex, so it spans everything, but includes things like absorption and lipophilicity and things that we'll also talk about briefly. So an example of a PROTAC is shown on the right. So this is an IRAK1 PROTAC, which is a part of a larger data set that's patented by J&J a few years ago. And it's because it's patented, I get to use it as an example throughout this talk. I will also be speaking about some internal data. I won't show structures, but I am allowed to talk generally about what we're seeing. So I will use IRAK1 as illustrative examples throughout. Okay, but first, to assess permeability experimentally, many groups use MDCK cells. So these are Madden-Darby canine kidney cells to assess the intestinal absorption of drugs. The problem is MDCK cells actually exhibit endogenous efflux activity, which leads to an underestimation of permeability. So to get around this, what we do and many other groups do is we use MDCK cells with transporters such as PGP or MDR1 are knocked out. And so those are used instead. So whenever I talk about correlations with permeability, this is the assay that we're getting our data from. And when we use this experimental permeability data from these knockout MDCK cell lines, we're able to train machine learning models that correlate pretty well with experiment. This is, so on the left-hand side of this slide in blue, you can see all the data for a series of PROTACs. And on the right in orange is actually just the set of compounds that the model had never seen and was synthesized after the model was created. So, this is in blue, actually, it's just the test set of all data, so 25%. And the statistics for that, Pearson's R 0.88, R squared of 0.77. The rank order is also quite good. And you can see that, we're able to get a pretty good understanding of how these PROTACs, at least in this specific series, so it's not necessarily generalizable to all PROTACs ever, but in this series, we're able to get a good idea of what the permeability will be, which is really exciting. And it's very helpful when we have large libraries of compounds that we need to select just a set to make. However, these are, what I just talked about, are ligand-based machine learning models. They're 2D, fingerprint-based models. And they're actually less useful for identification of what are actually the key contributors to permeability, especially if the contributors aren't just a single property or obvious descriptors. And that's usually what we find is the case. This is a complex issue. And from the industry perspectives paper that I mentioned earlier, some features that may be associated with PROTAC permeability that we have looked at and don't necessarily correlate perfectly with PROTAC permeability, but may contribute, are things like polar surface area, so a range of 100 to 200, molecular weight, less than 900 would be ideal, number of hydrogen bond donors and acceptors, and the presence of intramolecular hydrogen bonds. So you can see in the table over there, less than 14 hydrogen bond acceptors and less than 5 hydrogen bond donors. And lipophilicity also plays a key role, and to have a log D of around pH 7.4 of around 3 to 5 would be great. But there's also the question: does it go beyond just these descriptors, and is there also some component of rigidity or flexibility of the PROTAC? And so I'm bringing this up because one of the theories of how these PROTACs are actually able to permeate cells is that they exhibit some chameleonicity. So this means that in a membrane-like apolar environment, they're on the left in red, they assume this collapsed kind of state, so crumpled up, and in a polar environment such as the intracellular or extracellular compartment, they're in yellow, they assume a more elongated state. So beyond the ability, though, to transitions between the states based on the solvent environment, I also think the rate of transition probably matters. And that means, like, if a PROTAC is able to quickly adopt a collapsed conformation, get through a membrane, and then quickly adopt an elongated conformation in the right environments, it may lead to a better chance of getting in and out of the membrane, not getting stuck in the membrane, and also being able to attach to its E3 and POI binding partners and actually initiate that degradation sequence. So to assess this hypothesis, we created an unbiased molecular dynamics sampling workflow to determine the key conformations in different solvents. So we used water, methanol, and chloroform, which have dielectric constants of 80, 32, and 5, just about. And chloroform with a dielectric of 5 approximates the cell membrane. Obviously, water approximates the intracellular, extracellular environment, and methanol was somewhere in between. And first, we ran molecular dynamics simulations, which, as many of you know, integrate Newton's equations of motion to obtain a classical trajectory of how atoms move over time. And we ran about 4 microseconds of MD per PROTAC per solvent. MD simulations are great. They're fun to watch with some popcorn. But it's an immense amount of disparate data, especially as you scale up to not just 3 or 4 PROTACs, but if you're looking at hundreds, this becomes more complicated to interpret. So how do we condense this big data down into a human interpretable format, particularly in an automated way? So there are actually many such ways to do this. One is to use a Markov state model. It's a mathematical model that identifies kinetically relevant conformational states and the rates of transitions between those states. So very briefly, to build Markov state models, you perform some MD simulations, you cluster, and then use those clusters of states. So this sounds like a very normal clustering workflow. What does MSM theory add to this? Well, in the process of model refinement, we select for clusters which are more serious states. So they're so stable that things falling in must have forgotten where they came from before leaving. So we cluster these, assign these to states. And then after we define states, we can assess the probabilities of the ligand staying in that conformational state or transitioning to another state. So this method, allows us to identify the critical states of our PROTAC and as well as understand how the PROTAC moves between those states. So this is kind of the perfect application for this question we had. But the only issue is, classical Markov state models are really successful when many states are used, but because we're only interested in 2 states, collapsed and elongated, we used VAMP nets, which can directly produce an accurate model with very few states. So VAMP nets essentially replaces the MSM building pipeline of featurization, dimension reduction, MSM estimation, and MSM coarse graining with a deep learning neural network. We didn't come up with any of this. All the citations are at the bottom. This is just what we've applied to this project. And so once trajectories are discretized into the metastable states using Markov state models and analyzed with these neural networks, the results of this analysis, and the exciting part is that it provides the 2 states, collapsed and elongated. You can see collapsed that have in the salmon color and elongated in this teal. And I'll explain later on how the states are identified and what collective variables were used. But this is what we want to get out of this analysis. So we selected compounds with a range of permeability values to perform this analysis on. The data and everything I'm showing here is actually from our internal data set, so I can't share the actual structures. You can see a number of PROTACs here have a very low MDCK knockout value. A few of them have slightly higher, but this is by no means like very permeable compounds, but it is a range, and the range we are working with. But first we had to define what collective variables we were building the models on at all. So how do we define states? How do we define collapse versus elongated? So we already talked about a number of these features of PROTACs that might contribute to permeability or bioavailability. But what other characteristics can we actually pull from the MD simulations? So examples include, number of rotatable bonds or radius of gyration or RMSD. We actually ended up settling on end-to-end distance. And that's just measuring the distance from the furthest apart heavy atoms on the PROTAC. So a very simple metric. We tested, probably 24 of these different descriptors in order to try to find some sort of correlation that was generalizable across as many PROTACs as we could find. And what we found was that permeability is higher when the end-to-end distance is lower. So the plots below show 3 different compounds. They're all in chloroform. And these are just the MD simulation data. And the one on the left has the lowest average end-to-end distance and also has the highest permeability. So 18.5 angstroms end-to-end distance. So you'll notice that the distances are not very different, right? So the lowest is 18.5, the highest is 20.3. Like this is almost in the noise. However, the cool part about these 3 PROTACs is they also had very similar permeability values. So it suggests that even a slight change in collapsibility may affect permeability. So this is what we started with, and when we performed this analysis and looked at specific examples, pairs of high and low permeable compounds, we do see some differences. So what I showed previously was just the MD simulation data and looking at one descriptor. Now what we've done is we've done this MSM workflow, and we have the results. So I'm showing here a wonderful green blob, which unfortunately I can't show the exact structure of. But it has a higher, it has one of our highest permeabilities. And we find that in chloroform, there's a slightly higher percentage of collapsed conformation. By slightly higher, I mean 51%. So it's about even between collapsed and salmon and elongated in blue. And remember, chloroform is approximating the membrane here. But in contrast, when we look at compound 2, which notably is very similar to compound 1, except for this one R group, compound 2 has a much lower permeability. And in contrast, only 43% of the conformations are collapsed. So it's saying that in the membrane, compound 2, which has a lower permeability, isn't forming as many of those collapse conformations as the more permeable compound one. So we also know that intramolecular hydrogen bonds are associated with an increase in permeability. So in this paper shows that in apolar solvents, intermolecular hydrogen bonds form and allow for a more collapsed state with a lower 3D polar surface area. So when we looked at the formation of hydrogen bonds within the metastable states of the permeable and less permeable compound that I just talked about, we did find that the more permeable compound has more hydrogen bonds forming in its collapsed state. So that means that the more permeable compound is able to form those intermolecular hydrogen bonds, likely leading to a more collapsed state, where the less permeable compound isn't able to. And again, here are some more blobs that don't mean anything, except for that these represent the centroids that we're seeing in some of these metastable states from the Markov state model analysis. And we see that key intermolecular hydrogen bonds are forming in the collapsed states. So this isn't like revolutionary right here. I'm just saying that we are seeing hydrogen bonds actually form when we look at the collapsed form of the PROTACs. And then we worked with our experimental colleagues where we evaluated the same compounds as before in both water and chloroform. So we found that in DMSO, there are fewer hydrogen bonds formed than in chloroform, which is not surprising. Again, but it does make us more confident in our modeling results, which show the same thing, including actually the same intramolecular hydrogen bonds. So I'm going to walk through this figure with you. This is very complicated. Again, I've had to remove all the compound information, so I sketched it as best as I could. And the ABCD just refer to specific atoms, and the numbers there are temperature coefficients, and green means they're more positive, red means they're more negative. And the more positive temperature coefficient is associated with a stronger intermolecular hydrogen bond or increased shielding. So on the top there with the numbers, that's the NMR results in DMSO and CDCl3. The bottom is water MD simulation, a collapsed centroid, and bottom right is MD simulation, chloroform collapsed centroid. So what you can see here is that the intermolecular hydrogen bonds from NMR are present in the Markov state model ensembles, and that there are more intermolecular hydrogen bonds in chloroform and CDCl3 than in water, and so in both experiment and the MSM ensembles. And this gave us more confidence that what we're modeling is agreeing with experiment, meaning that we can use this information for without using NMR data in the future, and we can just use modeling and have some good confidence in this. And this generalized to a number of the compounds that we were looking at. It wasn't just this one example, but I figured you all didn't need to see more blobs of compounds. Okay, so this analysis was performed actually on shorter microsecond long simulations and clustered with just standard clustering methods. But interestingly, we didn't see any correlation with permeability, and we didn't see any of the intermolecular hydrogen bonds supporting the NMR results. And sure, those intermolecular hydrogen bonds appeared, but it was it didn't correlate with anything. We couldn't pull out any correlation. It didn't make sense. So it seems that sampling with longer MD timescales made a huge difference, and also interpreting the simulation data with MSMs provided more information for our analysis and compound design. So it seemed pretty necessary to be able to interpret that data. And so our ongoing efforts right now are we're using a data set of the 135 IRAC PROTACs and doing this workflow on all of them. And that's going to be publicly shareable, which is exciting. But we're also doing the same thing on the internal, similar number of PROTACs. And the nice part about that is we can, test it on 2 very different series and see for very different targets and see if this is generalizable. Notably, we're also working on improving the permeability score. So I didn't talk too much about that today, but what I meant by permeability score is we looked at these ratios of conformations and percentages of elongated versus collapse, and kind of a ratio of ratios. This is great. It would be nice if it was that simple. It's probably not that simple. It's probably much, much more complex. So, the first thing that we need to look at is, are two states really enough, collapse versus elongated? My guess is no. But, we're starting with the two-state theory and see how far we can get with it. We're also doing a comparison of all these matched pairs with high and low permeability. I just mentioned one today. And like I said, we're interested in doing some generalizability across other series as well. And another important aspect that I didn't touch on too much today is the reason we're so interested in permeability is because of the bioavailability. Ultimately, that's what the goal is. And we know that just because a PROTAC does not have high permeability, maybe it even has lower permeability, we do get compounds with low permeability to have bioavailability. So this is a complex issue. However, if you get into a permeable space, it can usually help get into a higher bioavailability space. So, how do we use all of this information to design PROTACs with higher permeability? Do we want to design in optimal intermolecular hydrogen bonds? And on that note, we only talked about intermolecular hydrogen bonds today, but there's so much data in the Markov state models that we can use to explore the conformational ensembles of these permeable and impermeable PROTACs. We're really just scratching the surface here. Okay, so with that, I just want to thank all of my collaborators at J&J. And in particular, I want to thank Anand Ota. He's a current research fellow at the Flatiron Institute. He was a 2023 J&J summer intern. He did his PhD at UCSD with Romy Amaro. And he is still working with us, and we have some, hopefully exciting results coming out in the next several months. So with that, I'd like to thank you for your attention, and I can take any questions.
Thank you so much, Bryn. I think this is a great presentation and really a good overview of your work in the product, on the product. So we have several questions on the chat. Let me start with the first one. Have you tried directly simulating the passage of products from the membrane?
Yes. No, so we haven't. We've thought about it though. So inducing, like pulling a PROTAC through a membrane, for example. We haven't done it. I'm curious to do it. I think it would be harder to do on a larger scale. I was hoping to scale this up to hundreds of PROTACs. I don't know how I'd scale the pulling through, but I guess we could. Yeah, so that would be a great idea. We have done some other work with other tools in membranes that didn't really work out too well, but anyways. Yeah.
Thank you. Now a question, permeability is different for the different tautomers. Did you consider this idea?
Permeability, sorry, could you repeat that? For the different tautomers, did you think?
Yeah,
I see. That's a great question. We have thought about this. We've also ignored it to some extent. That's definitely a scapegoat answer. But no, that's a good question. We basically haven't addressed it. Gotcha. Yeah, we understand that's an issue, or it's something to consider. But we have not worked on that particular issue.
For the tautomers. Okay. Yeah,
exactly.
That's fine.
Yeah.
The other question is, what is end-to-end distance? It's likely fully elongated structure. Have you done studies with molecular flexibility descriptors to correlate your results? Like, have you tried any molecular flexibility descriptors? to make a prediction for your
Yeah,
product?
so molecular flexibility descriptors. Do we have an example of like, I mean, are we talking about molecular flexibility in terms of like RMSD or radius of gyration or these sorts of
Yeah,
things?
do you have you tried any of those? Whatever
Yeah, we tried all of them.
all of
we
okay,
tried
good.
we tried several. We tried probably 24 different molecular flexibility descriptors. Basically anything we could think of. And this was like initial work, right? We were trying to correlate basically anything, any descriptor with to see if it would correlate. And end-to-end distance seemed the most reliable across all the PROTACs. So
Got you.
yeah.
We have another question. How could one determine if MD stimulation is low enough for MSM analysis?
Yeah, that's a great question. Depends on your system. And I think generally the more data is the better. I'm not necessarily the MSM experts, and there are many ways to analyze that. For this particular system, so we've worked with MSMs in grad school on larger proteins. For this smaller system, which is really just these aren't huge, they're large small molecules, but they're not giant. It seems that a couple microseconds is good enough for these. But like I said, so this is two different questions. Sorry, I'm getting into two different things. One is how long MD simulations need to be for Markov state model building. Just totally depends on the system, right? For this particular system, which I'm going to answer the question for, it seems to be that we need, more than a microsecond, even though that seems actually quite long to me. But it seems to get any of the information out that we want to. You do need more than a microsecond.
Gotcha. Yeah, one question I wanted to ask you, it's a little bit off topic, but the product have different linkers and different size of the linkers. Have you looked at that problem? Have you used any kind of like machine learning or in silicon methods to kind of like predict that? Because sometimes affinities depends from that too, as well as activity.
Yeah. The linker question. Linkers make a huge difference. You're absolutely right. We have looked at it in terms of, so we have very rigid linkers and very flexible linkers that are even the same length. So we were trying to control for length of linker by looking at the rigidity and the flexibility between the two. The flexible linkers allow for more compact PROTACs especially in chloroform and in water actually, right? They can fold up basically where the rigid linkers can't. We've looked at linker length, we've looked at linker type. Yeah, but just because a linker is very flexible doesn't necessarily mean it will be the most permeable either.
Okay, sounds
Hopefully
good.
That answers your question.
Sure. I have another question. So someone asked about the R-square of your machine learning models. It's pointed out 0.77, and they asked, did you try to improve that before you're trying MD simulation to get inside? So basically, yeah, I think it's a more general question. When you do sort of the machine learning and MD simulation approach in pile up in the same pipeline, have you tried to improve like the machine learning, or it's totally fine, like whatever the acceptable metrics for you in this case in terms of productivity?
We're always trying to improve the machine learning models for permeability. With this particular 2D model, the R2 score the R squared score is pretty good. It's good enough for our we have a cutoff basically where we have if it's predicted to be above a certain cutoff value, then we move it forward. If it's below, we get rid of it, and we don't move it forward. And we continuously challenge the model too. So at this model is pretty good, is basically what I'm saying. However, if you throw in something that the model has never seen before, something very different with very high permeability or something, and it's maybe a structure-based change that doesn't seem like it's going to be a huge difference, the model won't be able to predict that with very well. And we've seen that happen before, and that's basically why we pivoted to also using these physics-based methods. It's because you just can't capture it all with 2D information.
Sounds good. Thank you so much once again, Bryn, for the great talk and answer the question. Thank you.
Thank you, and thanks for the great questions, everybody.